Now let's look at the other side of the cell division, which is not the asexual reproduction, which is going to be based on sexual reproduction, and I think I've given you the name many times previously, and that is called as meiosis. Now, reproduction can also take place, for example, as a simple process which is called as budding. Budding is mainly when you have identical copy of a parent being given out, and it usually takes place in yeast cells. It's almost like a shoot that is coming out of the original cell, and it makes exact copies. It's almost like asexual vegetation. This is a very primitive form of reproduction. The most evolved form of reproduction is based on sexual means, and that is to do with meiosis. And again, I'm repeating, meiosis will only help to keep the chromosome number constant within each species. Meiosis also gives combination of alleles which are regions of a gene. Think of a characteristic of a gene that is being combined. So if a father has, let's say, blue eyes and the mother has green eyes, the variation that can come in the children is because of meiosis. And then none of your siblings and you would be exactly the same. Your children will not be the same. It's all because of meiosis, which gives a variation in a population. Very important cell division process. During cell division, usually gametes are formed. Gametes are either an egg or a sperm cells, and these are formed within the male and the female reproductive part, whether it's a plant or whether it's an animal. So if you're looking at a plant, we have the stamen, which is called as the male part of the cell of the plant, and the ovaries or the ovule, which is right in the middle, which is the pistil, is the female part. If you took a flower and you pull all the petals apart, you can actually see the male and the female parts of a flower. On the other hand, these are the two images that you can see from the human, and the top one, of course, is the male, and this is the female organs, and these are the different organs that will produce these cells. Now, after fertilization, if the chromosome number has not been somehow reduced, the fertilized egg will have twice its number of chromosome, and this is controlled by a process which is meiosis, in which the chromosome number will be reduced. The reduced number of chromosome is also called as the haploid form. The opposite of haploid is diploid. So diploid chromosome begins with the process of meiosis, which reduces to give the haploid chromosome number. Again, this is a repetition of what I just mentioned. What meiosis does is going to give you a fertilized germ cell, a germ cell that will have half or haploid chromosome number. So after fertilization, you have the zygote, which is the fertilized egg. So you can see over here, each of these cell divisions is starting with the germ cell, whether it's an egg or a sperm cell, and notice that the products of each are showing you one strand of the chromosome, which is half the number of the parental chromosome. This part is the main reason why we have meiosis, that fusion of two gametes will result in a zygote, the first new cell of an individual. So after fertilization, the first cell is formed that is called as a zygote. Meiosis also has the similar terminology. We will have the same terminology, but slight variation will be done in the first stage, which is prophase. In prophase, it's a very long process of prophase takes place during meiosis as respect to mitosis, and a lot of different things are happening. One of the common things that happens is the nuclear membrane will start breaking apart. The plasma membrane is still intact. The microtubule also start forming. Now, instead of the two chromosomes that were lining up in mitosis, now what will happen is homologous chromosomes will line up, which is, so if you have 46 chromosomes in humans, then 23 pairs will be formed. And it's because one pair is coming from the father side, one is coming from the male side. So notice that each of these units are actually showing you 4 individual strands. Each individual strand looks like there are 4 of them, but they're still in pairs. So this is called as a very unique process, which is the homologous chromosome are pairing up. And a second important feature that takes place is called as crossing over, where you can see the segments of these little strands are being exchanged. And when this takes place, when this exchange of the content of DNA takes place, and at the result, at the very end, these 4 threads will be separated, or the 4 chromatids will be separated, you will have variation in each cell that was formed. Remember, we had 4 cells over here. So if crossing over took place, then these 4 cells will be different. Back to my original slide. So you have this prophase, which is a very long process in meiosis. And the prophase begins pretty much the same. You have regions that are called as centrioles, and these centrioles will actually help to form the spindle. So they start moving around, and they will eventually go to the ends of a pole. Similar thing, nuclear envelope is breaking, and the third feature, which is the homologous pair that is happening. This is the different feature. This is the feature that is different from mitosis. Now here is an image that looks very similar to the first cell division. Metaphase. Why? Because you have the equatorial region of the chromosome being present. But the only difference is over here the chromosomes are not just 2, they're actually homologous, so there are 4 strands that are present at the equatorial region. The next phase of metaphase is followed by anaphase. Again, very similar. The chromosomes are going to be pulled apart, and at this stage the homologs are now being separated. So at the end of anaphase, you will have a duplicated chromosome giving one set towards one end and the other set towards the other part of the cell. Very similar to the previous image that we did. Again, telophase typically will divide the 2 nucleus, and you will have the nuclear membrane coming back, and the 2 segments of the chromatids are still intact within each part, but they have separated from 4 to 2 each. So this pretty much has reduced the number of chromosomes. Now, after telophase, usually previously we had a division of the cytoplasm, which will now not take place right now. This individual section that are 2 separated chromosomes will eventually go into the second phase of meiosis, in which again a prophase, a metaphase, anaphase, and a telophase will follow, and the segment of each are identical to what mitosis I was. So meiosis II is sometimes also similar to, or as is referred as mitosis, or just the mitosis I. So yeah, in which you will have exact same replica of mitosis, in which the chromosomes are in 2 chromatids, which are each strand. So this is one chromatid, this is another chromatid. Each is now forming a metaphase stage, in which you have them aligned in the center. Metaphase is then going to be followed by a similar anaphase. Again, the 2 chromatids are separated. You can see how it's separated, and you only have 1 strand each. Now notice over here that each of the chromatids are not identical. You can see how there are sections that look like they are they have exchanged parts, and this is what was happened in the first part of meiosis, which was meiosis I, in which in meiosis I within the prophase we have this section where the chromatids exchange parts. Now at the end of anaphase, you have separated the unduplicate chromosomes, and you can see that there are 4 sections of these nuclei, and these 4 sections are then now going to be separated by by this division of the cytoplasm to eventually give you 4 new cells, as you can see over here. So what has happened is you had a cell to begin with in which the homologous pair got together, and eventually through 2 subsequent steps, meiosis I followed by meiosis II, eventually separated into 4 cells. So if you had the first part of the cell in which let's go over here. You had the first part in which the homologous chromosomes were aligning up, were pairing up, then they aligned during the chromosomes and they separated out, and eventually you get the 4 haploid cells. And these 4 haploid cells are now going to give you 4 germ cells that will then fertilize with the other mate, and each cell has half the number of, just write down the words, half the number of chromosome. And this way, this has kept the chromosome number in its entire species. 2 events very important that will result in sexually reproducing species are chromosome crossing over of the chromosome during the first stage of meiosis, and also there's additional shuffling that also goes on in the metaphase that I really didn't tell you right now, but I'm just going to expect you to remember that crossing over or some exchange parts of the chromosome takes place in the first stage of meiosis, and this results in exchange of genetic material along its length. What does it result with? It results with new combinations of alleles that are not present in either of the parental generation, and thus results in variation of the daughter generation.